Hello everyone, till our previous classes, in our previous lectures, we have already discussed about the differences between apoptosis and necrosis. Second, the types of necrosis. So today we'll move further with the concept of mechanism, discussing the mechanism of apoptosis. Few things you need to remember here, right? Not many things, just few things you have to remember. One important thing is there are two pathways, right? Intrinsic pathway, extrinsic pathway. Intrinsic pathway like mitochondrial pathway, which is more important pathway. Second thing you have to remember, there are two phases, initiation phase and execution phase. Third thing you have to remember, the role of caspases. Caspases are very important. Whatever be the mechanism, whatever be the pathway, finally it is resulting in the activation of caspases, which are further leading to all the breakdown of cytoskeleton, endonuclease activation, fragmentation of nucleus. Everything here, whatever you see, is occurring after the caspase activation, right? So the role of caspases is very important. You should not forget that. Then next thing you need to remember is what is intrinsic pathway. See, intrinsic pathway or the mitochondrial pathway is the major mechanism of apoptosis. It occurs due to increased permeability of the mitochondrial outer membrane. In simple terms, you just understand that when a cell realizes that now it has to undergo death, right? A cell realizes that yes, I have to now die, right? So what will happen is, okay, so what will happen is, what are the factors which will make a cell realize that it should die now? Maybe the growth factor withdrawal, right? Growth factors are important for a cell to undergo growth and survival. You remove that growth factor, DNA damage, protein misfolding. So these can be the reasons which a cell will realize that it now has to undergo death, right? That will lead to, like, how will these changes be sensed? There are various sensors like ATM, right? So that will sense these withdrawals, growth factor withdrawal, DNA damage, and result in the activation of P53. P53 activation is a very important step, because then it will ask the cell to undergo death. How? By activation of pro-apoptotic signals, right? Simple terms, a cell has to undergo apoptosis or programmed cell death. So there has to be some pro-apoptotic molecules, there has to be some anti-apoptotic molecules. Now what do we want the cell to do? We want the cell to die, so we have to activate the pro-apoptotic signals, right? So P53 will activate the pro-apoptotic signals, right? Backs and back. Clear? So what will these backs and back do? These are pro-apoptotic. Okay? That means it has to do something which will allow the release of certain molecules, which has to make the cell undergo cell death or apoptosis. So what will it do? It will increase the mitochondrial permeability. It will increase the permeability of the mitochondrial outer membrane and allow the release of various molecules such as cytochrome C to get released into the cytoplasm. Released into the cytoplasm. So cytochrome C will get released into the cytoplasm and initiate the suicide program of apoptosis, right? Now this has to be tightly controlled with BCL2 family of proteins, right? So simply what you have to remember, when there is any withdrawal of growth factors, right, DNA damage, all this damage will be sensed by sensors in your cell like ATM, that will allow the activation of P53, and P53 will now activate the pro-apoptotic molecules. These pro-apoptotic molecules will increase the mitochondrial outer membrane permeability and cause the release of cytochrome C from the outer, this space, intermitochondrial membrane space, into the cytoplasm, right? And then it will activate the apoptotic pathway, right? So pro-apoptotic molecules, you have to, very important to remember the pro-apoptotic molecules, backs and back. So what will they do? They will increase the outer membrane permeability. How can it increase the outer membrane permeability? By forming channels in the outer mitochondrial membrane. So can you see, when the survival signals have collapsed, right, or there is irradiation, any severe DNA damage, misfolded proteins, these will be sensed, right? Activation of sensors like ATM, that will lead to activation of P53 and thus result in activation of pro-apoptotic molecules, and in turn it has to antagonize the action of anti-apoptotic molecules, because your body will have both pro-apoptotic as well as protective anti-apoptotic. Why are anti-apoptotic cells important, right? They will inhibit apoptosis when the cell is alright, when the cell is ready to undergo growth. It will inhibit the apoptosis. How? It will decrease, right? Anti-apoptotic molecules will decrease the permeability of mitochondria and thus decrease the release of cytochrome C, which will further inhibit apoptosis, right? Next thing you have to remember is the name of the anti-apoptotic molecule. So there is a very simple mnemonic, if you can remember, BCl2. As the name suggests, 2 means you are doubling something. Similarly, XL means you are increasing the size. XL is a larger size, 2 is doubling the amount. So these two things, BCl2 and BClXL, as the name suggests, want to increase the cell size, right? So they will inhibit apoptosis. So just a mnemonic that BCl2, 2 means doubling amount, XL means you are increasing the size, that means you do not want the cell to undergo apoptosis. So the anti-apoptotic molecules mnemonic is BCl2 and BClXL. As the name suggests, they will increase the size, they will double the size and thus inhibit apoptosis. How will they inhibit apoptosis? They will decrease the permeability of the mitochondrial membrane and thus decrease the transfer of cytochrome C into the cytoplasm. So once released into the cytoplasm, this cytochrome C will bind to apoptosis activating factor 1, which will lead to this hexamer or structure called as apoptosome, right? This complex will bind initiator caspase or caspase 9 and thus cleaves adjacent caspase 9 molecule, sets up auto amplification process. So simply remember, this cytochrome C will bind to apoptosis activating factor and form apoptosome, which will activate the initiating phase. So till now we are initiating the phase of apoptosis, right? So next we will see extrinsic pathway. What is extrinsic pathway? So these fast ligands will bind with fast associated via dead domain and finally lead to activation of the initiation caspases, right? So what you have to understand that finally, be it the intrinsic pathway or the extrinsic pathway, finally they will lead to the activation of initiating caspases and then executioner caspases, right? Right. So intrinsic pathway, what do you need to remember? The initiator caspase 9. Now just a mnemonic, can you see N, right? Intrinsic, so 9. It is a caspase 9, right? Extrinsic pathway is E, right? So it will be 8. Caspase 8 will be the initiating caspase in extrinsic pathway along with caspase 10, right? Then executioner caspases are common, be it the intrinsic or the extrinsic pathway. Finally, they are leading to activation of executioner caspases 3 and 6. Right? What will they do? They will degrade the structure and they will cause fragmentation of nuclei. They will cause cytoplasmic DNase activity. They will cleave the DNA. So finally, they are breaking down all the structures of the cell. What is important in apoptosis? You have to remember this is the programmed cell death where the cell will get converted into small bodies and finally engulfed, right? We want these bodies to be engulfed. So what do we want the cell to do? We want it to undergo fragmentation, right? So that these smaller molecules can be easily engulfed. Do we want the size to increase? No, we want the size to fragment. We want the nucleus to get fragmented. We want the cytoplasmic complex to get fragmented so that they are easily engulfed, right, by the macrophages. Right. So once these are fragmented, right, these cells will express certain molecules on the surface. Let's see, we have already died. You have to now be engulfed first. They will ask the macrophage to engulf them. What are they? They will first phosphatidylserine, right, that is normally present on the inner leaflet of plasma membrane, but in apoptotic cells they will flip out and get expressed on the outer leaflet so that they can be easily recognized the macrophages, so that the macrophages can easily phagocytose them, right? So these molecules are acting as as signals, like for the macrophages to recognize them and engulf them. Second is thrombospondin. These apoptotic bodies are coated with thrombospondin so that they can be easily recognized by the phagocytes. Third, apoptotic bodies are coated with natural antibodies and complement proteins, which are again recognized by phagocytes. So phosphatidylserine, thrombospondin, natural antibodies and complement protein C1q.